Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine your brain has its own dedicated cleanup crew. A tiny microscopic lymphatic system just working away silently in the background.
Yeah, clearing out waste, keeping everything, ticking over smoothly.
Exactly. And scientists have just found something pretty remarkable about how this system actually develops. And get this, it seems to be linked to something you're doing constantly. Just thinking, experiencing things, basically your neural activity.
It's a fascinating connection. Today we're going to dive deep into a new study that really sheds light on this.
Okay.
We'll be looking at how certain brain cells, these radial astrocytes specifically, work together with other cells to sort of orchestrate the growth of these really vital lymphatic vessels.
And these aren't like the lymphatic vessels elsewhere in the body, are they? Not like tubes?
No, not exactly. In the brain, it's more like a very delicate mesh, just one cell thick that forms right on the surface.
Interesting. So for this deep dive, we're looking at a scientific paper primarily.
That's right. The main source is a paper called Neural Activity Regulated and Glia-Mediated Control of Brain Lymphatic Development.
Okay.
And we've also got some context from a broader health news piece that talks about why brain waste removal is just so important generally.
Right. So our mission today is really to unpack this research, understand what it tells us about how the brain maintains itself.
Exactly.
So the study used larval zebrafish. Why zebrafish? What makes them suitable for this?
Well, zebrafish larvae are incredibly useful for this kind of work. For one thing, their bodies are transparent.
Okay, so you can literally see through them.
Pretty much. And you can also manipulate their genes relatively easily. So this allowed the researchers to actually watch in real time using microscopes how these specific cells were behaving and interacting as this lymphatic system was forming.
Wow.
Yeah, and the specific focus was on how the brain itself influences the growth of these meningeal mural lymphatic endothelial cells.
Meningeal mural lymphatic endothelial cells. Okay, that's a mouthful. So basically the cells that build these brain drainage channels.
Exactly, the building blocks of that surface meshwork we mentioned.
So what kind of techniques did they use to watch this happen? You mentioned microscopes?
Right, they used some pretty advanced methods. One key technique was in vivo time-lapse imaging.
So like making movies of the cells inside the living fish.
Basically, yeah. Taking images over time to see the development unfold step by step. They also use something called single-cell ribonucleic acid sequencing.
Okay, what does that tell them?
That's a really powerful tool. It lets you look at the genetic activity inside individual cells, so you can figure out, what specific job each type of cell is doing, what signals it's sending or receiving. It gives you a very detailed picture.
Got it. So using these tools, what were the big discoveries? What did they find out?
Well, one of the really key findings was identifying a very specific type of glial cell, a support cell in the brain. It's called an SLC6A11B positive radial astrocyte.
Okay, SLC6A 1B positive radial astrocyte.
And it turns out these particular astrocytes are the main source in the brain for a crucial signaling molecule.
Which one?
It's called vascular endothelial growth factor C.
The growth factor.
Exactly. And interestingly, these specific astrocytes have these long arms or processes that stretch right out to the surface of the brain.
So these specific astrocytes, the SLC6A11B positive ones, are they really important for building these lymphatic vessels?
Absolutely essential, it seems. The study showed pretty clearly they are indispensable for the growth of those meningeal mural lymphatic endothelial cells.
How did they show that?
Well, they use genetic tricks to reduce or even completely stop these astrocytes from producing that vascular endothelial growth factor C signal.
And what happened?
When they did that, the formation of that lymphatic network on the brain surface was significantly suppressed. It just didn't develop properly.
Right.
But conversely, when they boosted the production of that signal in those astrocytes, the lymphatic network development was enhanced. It grew more.
So it's like those astrocytes hold the on switch, or at least the dimmer switch, for building the system, using that specific growth factor.
That's a great way to put it. They have direct control via that signal.
And this is where the brain activity part comes in, isn't it? That was another really striking finding.
It really was. They found that neural activity, the constant electrical and chemical chatter between brain cells plays a big role in regulating how these lymphatic endothelial cells develop.
How did they test that?
One way was using a drug called tricaine. It basically dampens neural activity in the zebrafish larvae.
Yep.
And when they did that, they saw a definite reduction in the number of these lymphatic cells being formed. Less brain activity, fewer lymphatic cells.
Wow. And they also looked at sensory input, right? Not just drugs.
Exactly. In some experiments, they reduced visual input. They actually removed one eye from the larva.
Okay. And what effect did that have?
Remarkably, it led to problems with the lymphatic endothelial cell development, but specifically on the opposite side of the brain, the side that processes vision from that eye.
So it's a localized effect. Activity in one area affects the cleanup crew development right there.
It really seems that way. And even more telling, they did the opposite too. They increased neural activity, either by showing the fish moving patterns, stimulating their visual system, or even by applying gentle electrical stimulation directly.
And that caused more lymphatic cells to develop.
That's exactly what they saw, an increase in the number of these cells. So there seems to be this direct relationship. The more active a brain region is, the more it seems to invest in building up this lymphatic drainage system.
That is incredible. So a busy brain builds a better cleaning system for itself. But how does that link back to the astrocytes and their signal? How does neural activity talk to the astrocytes?
Ah, yes, that was the next crucial piece they figured out. The study showed that the way neural activity influences the lymphatic development is specifically through that vascular endothelial growth factor C signal produced by those SLC6A11B positive radial astrocytes.
Okay, so the astrocytes are the intermediaries.
Precisely. When they blocked the production of that growth factor in the astrocytes, then changing the neural activity didn't really have a significant effect on the lymphatic cells anymore.
I see. So the chain is neural activity tells the astrocyte, hey, we're busy here.
Mm-hmm.
And the astrocyte then ramps up production of the growth factor signal.
Which then tells the lymphatic endothelial cells to grow and form the network.
It's like the brain is managing its own maintenance budget based on how hard it's working.
That's a really neat analogy, yeah. It seems to have this direct communication line to regulate its drainage needs.
Okay, and wasn't there another cell type involved? Something about fibroblasts on the surface?
Yes, good point. They found another player, cells called calcium-binding epidermal growth factor domain 1 positive fibroblasts.
Right. Where are they?
These are located right on the surface of the brain, kind of sitting alongside where this lymphatic network forms.
And what do they do?
They cooperate with those radial astrocytes inside the brain to control the growth of the lymphatic cells. It's a team effort.
How do they cooperate?
What's really interesting is that these fibroblasts seem to be involved in processing the vascular endothelial growth factor C signal after it's released by the astrocytes. They help turn it into its final, mature, active form.
So the astrocyte makes the raw ingredient and the fibroblast finishes the job.
Something like that.
Yeah.
And this collaboration seems crucial for keeping the lymphatic growth restricted to the brain surface.
Preventing it from growing into the brain tissue.
Exactly. It acts as a boundary control. They actually showed this experimentally when they forced the production of the mature form of the growth factor deeper inside the brain.
What happened?
They saw abnormal growth of lymphatic-like cells invading the brain tissue itself.
Wow. So these fibroblasts are like gatekeepers, making sure the drainage system stays where it's supposed to be.
Precisely. Ensuring it works properly on the surface without disrupting things inside.
Okay, so putting all these findings together, the astrocytes, the growth factor, the neural activity link, the fibroblasts, what are the really big picture implications here? What does this tell us?
Well, stepping back, I think this is a really stunning example of how different body system, the nervous system and the lymphatic system aren't isolated. They actively talk to each other and coordinate their development.
Right. It's not just the brain existing and the lymphatic system cleaning up passively.
Not at all. It highlights a very active role for brain cells, specifically this astrocyte subtype, in actually shaping the brain's own environment, its immune niche, its cleaning capacity.
And this link to neural activity, that seems key.
Absolutely. That finding suggests this really dynamic connection between how much the brain is working and its ability to maintain itself, clear out waste.
So, does this mean perhaps that periods of high brain activity like learning something new or intense focus might actually trigger more lymphatic development or make the system work harder?
That's the intriguing possibility, isn't it? It suggests the system is responsive, adaptive, and might scale up its maintenance capacity based on demand.
And thinking longer term, could understanding this process help with brain diseases?
That's certainly the hope. I mean, if we can understand the precise molecular signals, the cells involved, how it's all regulated,
Yeah.
it could potentially open up new therapeutic avenues. especially for conditions where waste clearance in the brain is thought to be a problem, like neurodegenerative diseases. Alzheimer's is often mentioned in this context.
So if we could somehow boost this natural cleaning system.
Or restore its function if it's impaired, yes. That could be a potential strategy down the line. It's early days, but understanding the basic biology is the crucial first step.
Of course. And speaking of basic biology, are there limitations to this study we should keep in mind? It was in zebrafish, right?
Yes, and the researchers are very upfront about that. Zebrafish are a great model, and many basic mechanisms are conserved between fish and mammals, including us.
No.
But we can't just assume it works exactly the same way in the human brain, which is obviously much more complex. So further research is definitely needed in mammals to confirm these findings.
Makes sense. And do we fully understand how the neural activity signals to the astrocytes what the precise messages are?
That's another area for future research. While this study establishes the link, neural activity influences astrocyte signal production, the exact nuts and bolts aren't fully clear yet.
Like which specific neurons are talking to the astrocyte?
Exactly. What molecules are they releasing? What receptors on the astrocytes are picking up those signals? There's still a lot to uncover about the fine details of that communication pathway.
Okay. So what did the researchers themselves emphasize? What did they feel was most significant about their work?
One thing they really highlighted is how this changes our view of astrocytes. You know, for a long time, they were kind of seen as just generic support cells.
Right, the brain's glue or something.
Kind of, yeah. But this study, and others recently, are showing that astrocytes are incredibly diverse. There are different types and different regions doing very specialized jobs.
And these LC611B positive ones are a prime example.
Absolutely. Discovering their specific role as key regulators of brain lymphatic development really adds to this picture of astrocyte specialization and their active importance in brain health.
It really paints a picture of incredible complexity and very fine-tuned regulation happening constantly.
It really does. And the study also underscores this delicate balance the brain maintains. It needs to be protected, isolated to some extent.
The blood-brain barrier and all that.
Right. But it also needs to clear waste and manage immune responses. This whole system, the astrocytes inside, the fibroblasts on the surface, carefully controlling lymphatic growth, it's a beautiful example of that fine-tuned regulation in action.
It really is a testament to how the brain manages its own very specific needs.
Yeah, in a very sophisticated way.
So this discovery gives us a whole new way to think about this interplay between, our mental activity and the actual physical upkeep of our brain.
Mm-hmm.
It makes you wonder, doesn't it, how your daily routines, intense work, relaxation, even sleep, might be subtly influencing this cleanup crew.
It does. What questions does this spark for you, thinking about the brain's ability to manage itself?
Well, it makes me wonder about sleep, for instance. We know waste clearance ramps up during sleep. How does reduced neural activity during sleep fit with this idea of activity promoting lymphatic development? Maybe it's about function versus development.
That's a great question. Perhaps development is linked more to waking activity patterns, while the function, the actual pumping and clearing, is optimized during sleep's different activity state.
Yeah, or maybe different types of activity have different effects. It's clearly a frontiers, isn't it?
Yeah.
Still so much to learn. What research directions seem most exciting now?
I think confirming this in mammals is key, obviously, and really digging into those molecular signals between neurons and astrocytes, and maybe exploring if the system changes with age or in disease models. Lots to do.
Definitely. Well, that was our deep dive into how neural activity and specific glial cells control the development of the brain's lymphatic system.
We unpacked the fascinating role of these specialized astrocytes, their signals, and how brain function itself shapes this crucial maintenance network.
We really hope this exploration has given you a valuable shortcut to being well-informed on this exciting piece of research.
Join us next time for another deep dive into the world of scientific discovery.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.